taking the last run towards finishing this particular course, and that is blood banking and transfusion medicine. Now, this is going to get you a couple of important questions from point of view not only of path but also medicine. I'll start with the basics. I'll tell you about the blood groups, the methods of blood grouping, blood donation, blood bags. We'll wrap it up right in the end with a couple of vacutainers also that are important. So let's get going. The first thing that you need to know is about blood group genetics. So now the blood grouping system that we follow, the AB blood grouping that we have, was given by Landsteiner. Landsteiner was the scientist who gave all of that to us. So I want to start off the story by talking about something known as H gene. Then I want to talk about the usual A, B and O genes that all of you know. And then we'll talk about the RH gene that of course is again very, very famous. So when I say H gene, that is going to be present on chromosome number 19. Then the A, B and O are going to remove the 1, keep the 9. So that is going to be chromosome 9. And the RH gene is going to be chromosome number 1. So that is how I have learnt it, that when it comes to 19, the first gene, actually, I will now tell you a pathway in which I will tell you that the H gene is going to be the primary gene from which everything is going to rise. So I've given the biggest number to it. Then I'm going to split the 19 into 9 because the next most important is A, B and ABO wala blood groups. And then the 1 is going to go to the RH group. So that these are the genetics, these are the chromosomes that you have. Now you're going to move forward and talk about this H gene that I'm talking about. What does this H gene do? Please note that this H gene is going to be a kind of a fucosyltransferase. This H gene does the work of fucosyltransferase. So break the word. Transferase, what is it going to transfer? It is going to transfer fucose. It is going to transfer fucose. So what happens is that there is something that the H gene makes. The H gene makes something called H substance. This H substance is now going to be added like a fucosyltransferase. If it is going to add a fucose molecule, it can result in the formation of the A blood group. So for example, you don't have to necessarily learn this, but if it adds a fucose molecule on N-acetyl galactosamine, if it acts a fucose molecule on N-acetyl galactosamine, it is going to make it into an A blood group. If it adds the fucose to what? If it adds the fucose to galactose alone, it is going to become the B blood group. If it is going to add the fucose to both N-acetyl galactosamine and galactose, then it is going to be making it a AB blood group. And if it is not going to add any fucose to any of them, it will become an O blood group. So simple. It can either make an A blood group, or it can make a B blood group, or it can make an AB blood group, or it can convert into O blood group. So this is a conversion. So imagine H substance is converting into only one thing over here, that is A blood group. H substance is converting into only one thing over here, that is B blood group. H substance is converting into two things over here, A and B blood group. So maximum conversion is happening. H is maximally going to convert over here. And in O blood group, H is not converting at all. So if I ask you, where do you see the maximum H substance? Which particular blood group? You will say, ma'am, that blood group where H has not converted at all. And that is going to be blood group O. And where do you see the least H substance? You will say the place I will see least H substances where all of the H substance has converted into something else. And where is it converted maximally? It has converted into the AB blood group. So life is very simple. But in blood banking, they don't keep it simple. They say that for testing of H substance, remember this very carefully for the higher institute exams, testing of the H substance is going to require something known as anti-H antibodies. And what is anti-H antibodies? It is obtained from a plant known as Ilex lectin. Ilex europaeus lectin. You don't have to learn the entire name, just learn Ilex lectin is going to give me anti-H. I will add few drops of anti-H and I will find out that how much of H substance is present in a gene. So on doing that, they realized that the maximum H substance was present in blood group O and the least H substance was present in AB, which I have already told you, because in AB all of it has gotten converted into AB, whereas in O there is no conversion, so maximum H substance is there. But you will realize that the pathway in between is a little complicated, because we know that I don't just have A and B blood group. A blood group people further have different types. There are some people who have blood group A1, there are some people who have blood group A2 and so on. So remember, from maximum H substance to least H substance. So O has maximum, then there is A2. So एक A वाला blood group हो गया, then let me take a B वाला blood group, then let me combine the two, then let me write A to B. So A2 is done, then B is done, then the A to B. So A2 का पूरा story is done. So now I can give importance to A1 and then I will give importance to A1B. So let's do a complete this thing. From O, first I'm going to start with A2. After A2 there is B, then combine the two, that is A2B, then there is A1, then there is A1B. That is where how it goes from maximum to minimum or the least amount of H substance that is present. How do you test for H substance? By using Eulex Europeus lectin, that is anti-H. Now let me show you the table, which is one of the most important, but you studied it since your NEET UG days also. How do you classify blood group A, B, AB and O? What are the antigens? So you all know that whatever I'm going to tell as blood group A means what antigen does it have? The name of the blood group is the A antigen. A blood group matlab A antigen, and the antibody is going to be the opposite one. So this patient will have which antibody? This patient is going to have the anti-B antibody. So repeating, blood group A means A antigen and the antibody is going to be anti-B. When we talk about blood group B, means the antigen is B. So this time the antigen is B and the antibody will be the opposite in the patient. It is anti-A antibody. When I say there are both the antigens present, blood group A and B hai, AB hai, so this means both the antigens are there. AB, both antigens are there. And if both antigens are there, then the person cannot have any antibodies. Antibodies are none. In O blood group, zero antigen, anda. O anda, zero antigen, and when no antigen is there, this means all the antibodies will be present, anti-A and anti-B. So simple, the name of the blood group that you have is the name of the antigen, and the opposite situation you will be seeing in the antibodies. Let me tell you, and I think all of you know this for a fact, that the most important part that I've already told you is that whether you're dealing with blood group A or blood group B or AB or O, one antigen which is common to all, which I've not written separately, one antigen from which the entire story started, which was converting or not converting, something that is present in all is going to be H antigen. And if all of them have H antigen, none of them will have any antibody. Why am I telling you that? Because now I want all of you to be able to analyze something called Bombay blood group. Firstly, it was identified in Bombay, so that is why they called it Bombay blood group. It was identified by a group of scientists called Bende et al. So these are the names given to it. What is Bombay blood group? You will say Bombay blood group is a person who does not have A antigen, who does not have B antigen, who does not have any antigen. मतलब A भी नहीं है, B भी नहीं है. You will say, ma'am, if this person doesn't have any antigens, this means he will have antibodies. Means he's going to have anti-A antibodies, he's going to have anti-B antibodies. Isn't that looking like O blood group only? Not having any antigens and having all antibodies, this is looking like a O blood group. Why are you calling it Bombay blood group? Because O blood group people at least had H antigen. Na? H तो is there in all. Bombay blood group people don't even have the H antigen. That is what is different. H antigen is not there. This means there is going to be anti-H antibodies present. So when I talk about Bombay blood group, I always mention that Bombay blood group is something that is going to have no antigen at all. There are no antigens, and when I say no antigens, A, B, H, everything is missing, and all antibodies are going to be seen. Anti-A, anti-B, anti-H, all antibodies are there. I hope you've understood the difference between a Bombay blood group, as well as an O blood group. O blood group में भी antigens नहीं है, no A antigen, no B antigen, but at least H is तो there ना. In Bombay blood group, even that H is missing, even that H antigen is missing, right? Now there are some minor blood groups also. The ABO story to is fine. So let me ask you some questions. Who is said to be the universal donor? Think logically. Why is there a universal donor? Universal donor will be someone who does not have any antigen. All the antigens are missing. So all the antigens are missing. That person becomes a universal donor. Who is going to be a universal recipient? Means that person does not have any antibodies. So you can give him any blood and he will not attack because there are no antibodies. So he will receive any kind of blood. Universal recipient is AB. But this is, when they are talking about case of, say, you have an emergency situation. Yahan par, there's a discrepancy which you have to look out for. There's an emergency situation. Patient has met with a road traffic accident. He is bleeding profusely and you don't have any time to give him, to get his blood group and cross match and all of that. He's bleeding. He will bleed to death till the time your blood group report will come. You will have to give him blood stat. What is the safest blood group? that you can give to him. Who is the universal donor? Who is going to be, which blood group will you transfuse? Most importantly, you are going to transfuse O blood group because it does not have antigens. Now you are going to give O blood group. However, whenever I'm saying these situations, I'm talking about whole blood or packed red blood cells. In case they ask you that in a case of emergency, you want to transfuse plasma. See, plasma has what? You want to give a patient plasma. Which kind of plasma will you give a patient? A plasma that has all the antibodies so that it should go and kill the patient? No. You will give that person some innocent plasma which doesn't have any antibodies. Which is the plasma which doesn't have any antibodies? AB ka plasma doesn't have any antibodies. So if they just ask you which is the universal donor and which is the safest blood group to give in an emergency, means they've not mentioned red blood cell hai, you assume red blood cell, then you write O is the universal donor, I will give O. But if they mention specifically which plasma will you give in case of emergency, that plasma which is innocent, it does not have any antibodies, and that plasma is going to be the AB blood group plasma because that does not have any antibodies. Done with that. Now we are going to start with minor blood groups. There is a blood group known as the Lewy blood group. Means this is something to do with the Lewy antigen. You know, please remember this antigen is of course going to have some antibodies against it. IgM type of Lewy antibodies can be there. It's a very minor blood group. If you want to know some extra information, please note that this particular antigen is not going to be synthesized by red blood cell. This is Chori ka antigen. Lewy is the liya hua antigen. Liya hua antigen means I am not making this antigen from my plasma, I am going to absorb it. This is basically going to get absorbed from the plasma. This is going to get absorbed from the plasma. So repeating once again, what is this Lewy antigen? It is liya hua antigen. It is the antigen that I have not made but I have picked up from the plasma. That is what is Lewy antigen, and what is the antibody against it? It is the IgM antibody. Then there is another antigen known as the Kell blood group. When I am talking about the Kell blood group, these are people, you know, who have this blood group, but sometimes there can be deficiency. So there is a Kell phenotype deficiency that can be noted, and that is known as a McLeod phenotype. So please remember, I'll write it like this. If there is a Kell deficiency, that is referred to as the McLeod phenotype. And what is McLeod phenotype also known as? This is referred to as neuroacanthosis syndrome. What is neuroacanthosis, guys? Break the word and you will get the meaning. When I say neuro, there is going to be neurological deficit. There is going to be neurological and muscular deficit. So there are muscle problems. You will see there is some muscle dysfunction. There is weakness. There is dystrophies that will happen. And acanthosis means that what will you see in the blood vessel or the blood cells? You will see acanthocytes. Do you remember from your discussion of acanthocytes in way back in your RBC chapter? Acanthocyte had those spikes. Yes, I can see these red blood cells with spikes. So I'm seeing acanthocytes, and that is a McLeod phenotype. McLeod phenotype says that there is neuroacanthosis. Neuro means for neuromuscular problems, and acanthosis means there's going to be acanthocyte. That is the Kell blood group. Not present, McLeod phenotype. There's another blood group or an antigen called the Duffy antigen. So please remember, Duffy antigen is under the control of the dark gene. DD wala ho gaya. It is under the control of the dark gene. And if this is a red blood cell and this red blood cell is going to have the Duffy antigen, you know what is Duffy used for? Duffy antigen is used for the attachment of Plasmodium vivax and Plasmodium knowlesi. Knowlesi, where have you studied Plasmodium knowlesi? Vivax, so you've read a million times. Where have you studied Plasmodium knowlesi? Way back in your in microbiology, I've taught you under parasitology section. Plasmodium knowlesi is seen in Malaysia. It came from the monkeys. It was the wala Plasmodium. It came from Malaysia, from the monkeys, right? So these two things enter via Duffy antigen. That means that if people have Duffy blood group or Duffy antigen negative, will Vivax and knowlesi ever be able to enter? No, and that basically means there is going to be a protection against Plasmodium vivax and Plasmodium knowlesi. If Duffy is not there, they won't be able to enter. As easy as that. What all antigens have you done? You've done the main A, B and O. You've also spoken about their absence in Bombay. You've spoken about Lewy antigen, the Duffy antigen, the Kell antigen. Now I'm going to take you to some antigens which you have definitely studied again in RBC chapter. If you remember, every RBC has something called the P antigen And then every RBC had something called the I antigen. And where did you study these? In autoimmune hemolytic anemias. In autoimmune hemolytic anemias, remember I gave you a mnemonic called Mike? What was Mike? Mike was that when you are dealing with cold autoimmune hemolytic anemia, cold means which antibody? You are dealing with IgM, and IgM is against which antigen? IgM is against I antigen. So when antibodies will come and attack the I antigen, this is cold autoimmune hemolytic anemia. And for the P antigen, obviously then it becomes warm autoimmune hemolytic anemia. The warm, garam, IgG antibody was coming here, hai na? One more question about P antigen that you get, which you have studied in microbiology. Parvovirus B19. Parvovirus B19 goes and attacks which red blood cell antigen? It goes and attaches to the P antigen of the red blood cells. These are all the P and I antigen questions you can ever get in path, in micro, in medicine, repeating. The Mike will remind me that in cold autoimmune hemolytic anemia, it is the I antigen and the IgM antibody. So automatically the P antigen becomes in warm autoimmune hemolytic anemia. The next thing that you have to know is that parvovirus B19, also P4P, attacks the P antigen of the red blood cells. So now all these H antigen story, ABO antigen, minor blood groups are done. Now I can come down to the RH antigen. What chromosome was it on? It was on chromosome number 1. And there are going to be multiple genes. There is a C gene, the D gene and the E genes, or the antigens which are very, very common, out of which the D1, out of which the D gene or the D antigen is the most common. That is why you must be hearing many times they say that we have added anti-D antibodies to find out someone's blood group. What is anti-D antibodies? Anti-D means they are basically trying to find out the RH status. RH is recommended as what? You know, patient is said to be positive or negative. So this is not given in D, C and E. This is given as AB positive or AB negative. Positive and negative is how the reporting is done. But the most common antigen that we indirectly target is going to be the D antigen. The story is that when we were talking about the ABO system versus RH system, you need to know a slight variation between the two. What all have you studied? Have you done the genes already? Have you done that ABO blood group was on chromosome 9, RH is on chromosome 1? Have you done ABO blood group is going to be blood group A, B, AB and O, whereas RH is going to be positive or negative? Ye ho gaya. Their antibodies, please remember. ABO blood group is IgM antibody, means it is not going to cross the placenta, means that makes sense na. So many times you've seen that the mother is A positive and the baby is B positive, but they don't start fighting with each other in the womb. Why? Because AB blood groups are to do with IgM antibody. IgM antibody does not cross the placenta. On the other hand, when you look at RH antigen is to do with IgG antibodies. This means this can definitely cross the placenta. And when I say this is going to cross the placenta, that basically means that if the mother by chance is RH negative and the baby is RH positive, or if it's an opposite scenario, basically an RH incompatibility can create an issue. RH incompatibility in pregnancy does create, RH incompatibility hota hai, does create an issue. Why? Because these are IgG antibodies and that can cross the placenta and can cause problem to the mother and the child. Hai na? So remember, this is what you had to know regarding their antibodies. Finally, where are these antigens most commonly expressed? So remember that the AB blood group antigen, they are seen in saliva. The antigens are they are called as secretors. Secretors means you see them in the saliva, the semen, the sweat. You don't see them in CSF although. Okay? CSF mein nahi, but saliva, semen, sweat mein you see it. RH antigen is not going to be present in any of these body secretions. So the body secretion thing is definitely going to be the AB blood group secretors. They are called as secretors. Okay. Now you have done